Good evening. I'm Chris Bolzan, Executive Director of the Gloucester Marine Genomics Institute, and it is my pleasure to welcome you to the second session of the GMGI Fall Science Hour series. Last month, we kicked off this season with an exciting talk by Dr. Bruce Walker of the Reagan Institute, and we are thrilled to share a new lecture with you at home tonight. We have a record number of guests today and a great many new registrants, and we are so glad that you're here. Tonight is exciting as we welcome Dr. Bill Kaelin, the 2019 Nobel Prize winner in Physiology or Medicine from the Dana-Farber Cancer Institute. It is such an honor to have him, and he will be introduced and joined by his friend, GMGI co-founder and board member, Dr. Mark Vidal. And it is also exciting for us to have so many of you online with us. We appreciate the great feedback, ideas, questions, and enthusiasm that we have received for this series. And we are so encouraged by the tremendous appetite for science education shown by our community. When GMGI was founded in 2013, local scientists and entrepreneurs envisioned that the ocean could be a renewed source of opportunity for Cape Ann. Our ambitious mission is to address critical challenges facing our oceans, human health, and the environment through innovative scientific research and education. By bringing world-class science and transformative workforce development to Gloucester's historic waterfront, GMGI is catalyzing the regional economy. Recently, as the science community has come together like never before, we have seen many new ways in which the founder's vision has become reality. Our Gloucester Biotechnology Academy graduated its 4th class, and last week, our 5th class successfully, in person, and safely completed semester 1. Our graduation and career placement rates consistently outpace vocational and community college norms. In our research institute on the harbor, our science team, led by the Donald G. Combs Science Director, Dr. Andrea Bodner, conducts marine research powered by genomics. And this year, they cracked the lobster genome and also established exciting new strategic partnerships with collaborators, including OceanX, among others. You'll all have an opportunity to hear from Andrea during our December Science Hour, when she will share her work with sea urchins as models for living long and healthy lives. We made headway in 2020, growing the science community on Cape Ann, fueled in part by the work of GMGI's entrepreneur in residence, Dr. Eric Huang, and evidenced by a recent study that there are now nearly 40 life science and biotech companies operating within 17 miles of our headquarters, many of whom are employing our academy graduates and bringing new life to the North Shore economy. Though we all continue to navigate the challenges brought by the pandemic, we are working hard to try to bring you good news, and we are profoundly grateful for your support, your outreach, and guidance received in recent months. A special thank you goes out tonight to the following organizations and individuals who sponsored this evening's event: the 1911 Trust, a wealth management firm on the North Shore focusing on wealth management for generations; the James and Gail Bacon Family Trust; and New England Labs, with expert knowledge in design and installation of laboratory furniture. They're currently partnering with us on the biomanufacturing expansion of our academy. We are thrilled to have Nobel Laureate Dr. Bill Kaelin as our speaker this evening. Dr. Kaelin will share with you his remarkable journey to Stockholm. And I'm going to turn the screen now over to our GMGI co-founder and board member, Mark Vidal, who's going to introduce Dr. Kaelin and facilitate the Q&A. So if you have questions you'd like to submit, please use the Q&A button at the bottom of your screen, and we're going to do our best tonight to answer all that time allows. So with that, enjoy everyone, welcome, and over to you, Mark.
All right, well, thank you very much, Chris, and many thanks to all of you for being here with us tonight, taking the time, and also, of course, for all the support that you gave us, as Chris reminded us of. And of course, Bill, thank you very much for taking the time to do this. So it's a really great pleasure to introduce my colleague and friend, Bill Kaelin. He's the Sidney-Farber Professor of Medicine at the Dana-Farber Cancer Institute and at the Brigham and Women's Hospital. He's also an investigator of the Howard Hughes Medical Institute. So while Bill and his lab were investigating in the late 90s to early 2000s how a particular gene, the name doesn't matter, works or doesn't work actually in cancer, they made really a stunning discovery, stunning. So they identified a molecular mechanism that allows the cells in your body to sense oxygen. So, you know, 3 words: body, cells, oxygen. It's very simple, right? So it doesn't take a huge amount of science background to understand how important this was. I mean, think about how critical oxygen is in our lives. And yet 20 years ago, before Bill's discovery, we didn't really know how that worked. So as Chris mentioned already, Bill received the Nobel Prize in Physiology or Medicine for this work, and of course, many other awards. He's a member of very prestigious academies, like the National Academy of Medicine, the National Academy of Sciences, and pretty soon, the Gloucester Biotechnology Academy. Just kidding, Bill. Yes, let's cross fingers. So Bill, and I'll finish with that, also cares really deeply about several societal issues, not the least of which is education and training, and so of younger generations. And so I'm sure he'll be really a great inspiration to our Academy students, who I salute tonight, by the way. Hi, guys. And who are here tonight to listen to him as well, in addition to everybody else. So Bill, as Chris mentioned, it's an honor to have you tonight as a GMGI Science Hour speaker. Warm thank you for accepting to do this. Take it away. Welcome to K-Pen.
Thank you very much. And hopefully we can even get my slide sharing to work, which is always step 1. Okay. And hopefully you can see my slides.
Yeah.
Okay. So I thought I'd start with a few comments about my childhood. Let me see if we can pick up a pointer here. Let's get a laser pointer. Let's make it green, and then we'll be good to go. So I had very devoted parents growing up, but I would say they are not what are sometimes called helicopter parents. I really had remarkable teachers, but I feel a little bit guilty about this because some of my female teachers, sadly, you know, grew up in an era where a lot of women were told their career options were to be a nurse or a secretary or a teacher or maybe a nun. And I'm quite sure some of my teachers back in the 60s and 70s today might have been doing other things, but I'm glad they were my teachers. I had ample unstructured time. I think that's important. And I had a lot of toys which sort of directly or indirectly fostered an interest in scientific and in science or engineering. I had a microscope, I had a chemistry set, an erector set, electric cars, et cetera, and of course, many of you will know this was not an accident because I was born in 1957, and anyone who was a child in the 60s grew up in an era where science and engineering were celebrated by politicians and other stakeholders, because of course we were in the middle of the we were in the middle of the Cold War, which in turn beget sort of the space race. And so there was bipartisan support for science and engineering, and the language coming out of Washington, again, celebrated science and engineering. And so like a lot of young boys in the 60s, one of my heroes was Neil Armstrong, but I grew up on the South Shore of Long Island, so I will admit that I thought Joe Namath was actually far cooler, which is maybe a good segue then to some of my study habits. So when I was younger, I would say about myself that I liked learning concepts and ideas. I liked objective subjects that had a right answer or a wrong answer, and I liked playing. On the other side of the ledger, I disliked memorizing facts. I didn't like subjective subjects, and I didn't really like studying, to be all that honest. And so as a result, I was sort of a mediocre student. So even through high school, I was mostly a B student, except thankfully in math. So in math, I usually got A's because math, you didn't, if you were lucky and I was lucky, you didn't have to study. It was very conceptual. There was no rote memorization, and thankfully it just came easy to me. So despite my abysmal study habits, I always sort of got A's in mathematics. Then something changed my life. So this was now 1974. My high school was one of the first high schools to get a remote computer terminal. In this case, it was linked to a mainframe at Fairfield University. And this was sort of exciting to me because computer science seemed like maybe, you know, math come to life, that you could actually use mathematical thinking and a mathematical approach, but now do all sorts of things with the power of the computer. So this was very intriguing to me. But perhaps more importantly, one day I looked in the waste paper basket next to this computer terminal, and there was a pamphlet that was recruiting young students to attend an 8-week summer program at Florida Atlantic University. So this was a National Science Foundation, so-called student science training program, where 32 high school students were going to come and study college-level mathematics and computer science. So sort of on a whim, I applied, and somehow I got in. Again, fortunately, I had always had good math grades and had done well on math aptitude tests. So I guess they overlooked my many other deficiencies. So I was accepted, and here I am. I actually went to this summer program, and this actually completely transformed my life for a number of reasons. So now, for the first time, I was just surrounded by these incredibly smart kids, and the curriculum now was challenging, and it was interesting, and I really got rather excited for the first time about school. And so one of the lessons I learned here was to try to surround yourself with people who are smarter and more talented than you are. So it might feel good for your ego to always be the smartest person in the room, but if you're always the smartest person in the room, you're probably not learning as much as you can. So I've taken this simple truth with me to try to surround myself by people who are smarter and more talented than me. Now, I went down to this program with some trepidation because I heard about these, you know, other 31 terrific students, but I was actually pleasantly surprised to find out I wasn't the dumbest of the 32 students. In fact, I could actually hold my own pretty well, but they all had much, much, much better grades than I did because they all had much, much, much better study habits than I did, which was admittedly not saying very much. So I decided to do an experiment. In fact, arguably, maybe this is the most important experiment I ever did. So I decided for my senior year, I would do an experiment. I would sit in the front of the classroom instead of the back. I would spend more time with the smart kids, also known as the nerds, rather than sort of poking fun at them. I would actually do my homework. That was a revolution. I was actually going to do my homework and try to get that done as the first thing during the day after I got home from school, rather than the very last thing I would possibly tackle. I would review the day's new material every evening, and I would do extra work rather than the bare bones minimum. And so this experiment actually worked. So now I did very well during my senior year. In fact, I was so proud of myself, I saved my report card, which I wasn't going to show, but in looking at it, I was amused that the one course I couldn't get an A in apparently was trampoline. So who knew? So maybe that's the blemish during my senior year. Okay, so now I'm sort of a late bloomer. Somehow I pulled off a good senior year, and I have pretty good aptitude scores. So I actually did get into MIT, and I was very proud of getting this letter and being told that I was accepted to MIT. But at this point, what you have to know is that my father was the first member of his family to ever go to college. And he had worked his way through school, and he had gone to Duke University, and he was fiercely loyal to Duke. And his dream was that his eldest son, namely me, would go likewise to Duke and wouldn't face the sort of financial hardships that he had faced as a young boy in college. So I remember the day I walked into the den with this acceptance letter, and I told my father, you know, Dad, I really think I want to go to MIT. And at that point, he put his scotch down and he said, don't you have to be really good in math to get into MIT? And I said, yes, sir. And so he said, well, I'm going to give you a math problem. If you go to MIT, you'll pay your tuition. But if you go to Duke, I'll pay your tuition. Well, this was a pretty easy math problem. So I enrolled at Duke University, and true to his word, he actually did pay the tuition. I think you can see that there's been some inflation in tuition and room and board cost since I went to school in the 70s. But I did go to Duke, and I got a great education there. Now I have to start thinking about what am I going to do for a career. So I told you my first loves were mathematics and computer science, but I'm embarrassed to admit I certainly was no Steve Jobs. I wasn't a Bill Gates. I did not see the personal computer revolution on the horizon. And I was really worried that if I pursued computer science, I'd be working in a back room someplace, writing code for industrial and defense applications, and that didn't really appeal to me. So I thought about what else could I do? So again, I like math and science. I wanted to be my own boss. I wanted new intellectual challenges every day. I actually preferred a job where I was interacting with people. I thought it'd be nice to have a decent income to support my hopefully future family. And I wanted something where I felt like I was going to be helping others. So I decided to become a pre-med in my focus. And so I pursued a pre-med curriculum at Duke, although I will say I also did major in mathematics, given my first love. So I actually did quite well at Duke with these newfound study habits, but someone whispered in my ear, you know, if you really want to go to medical school, it would really look good if you worked in a laboratory, just to sort of round out your application. So I met with a Duke chemistry professor about working in his laboratory on an independent study project. And I remember him telling me that he had had other undergraduates work on this particular project in his lab, and that all 7 of the people who had worked on this project had gotten into medical school. So that's all I had to hear. I thought this was now the golden ticket to get into medical school. Now, of course, I was only 21 years old at the time. If I was a little wiser, I might have asked, well, why couldn't these 7 undergraduates who'd worked on this project previously, why could none of them bring this project to completion? And so I joined this laboratory and I became the 8th person who could not bring this project to completion. And with the benefit of hindsight, I can tell you everything that could be wrong was wrong. This project was uninteresting, unimportant, and undoable. My mentor rarely, if ever, interacted with me. No one in the laboratory was doing anything remotely similar to what I was supposed to be doing, so no one could help me. And then I had the audacity during my last lab meeting to correctly point out that this project really was based on an artifact or a mistake that had occurred many years ago and would never be brought to completion. So he rewarded me by writing on the margin of my transcript, Mr. Kaelin appears to be a bright young man whose future lies outside the laboratory, which is like being told you have a good face for radio. And he made sure that on that very same transcript, I was given a C-, which of course for a pre-medical student is almost like having a wooden stake driven through your heart. Now, despite this, I actually did, well, I should say, first of all, the C- had its intended consequences on many of my medical school applications. So here's, for example, my application to Harvard Medical School. But fortunately for me, 2 medical schools decided to take a chance on me. One was the University of Connecticut and the other was Duke. So I enrolled at Duke Medical School. You can tell this is the 70s from the hair here. And so I started off again in medical school thinking I'm going to be a clinical doctor. But the Duke Medical School curriculum was a little bit unusual in that in the 3rd year, you had the option to work in a laboratory. And so with considerable trepidation, I again set foot in a laboratory, now working for Randy Jerdal. And I was studying tumor blood flow, how tumors obtain a blood supply and how that blood supply can be manipulated with drugs such as, in this case, verapamil. So this was a much better experience, but it wasn't enough to convince me to become a scientist, frankly. But I point this out in part because you can see here the subject of this paper is tumor blood flow, and I actually did learn a lot about so-called tumor angiogenesis during this time period. I should also say, I sometimes tell young people, the most important thing for a young person entering the lab is to have something, to a first approximation, anything work in their hand. So at least here I did have something work. It did lead to a paper, but again, it wasn't quite enough to make me want to be a scientist. But I did, as I was about to say, learn a lot about tumor blood flow and tumor angiogenesis. And I've been impressed over the years how many times I've learned something in one context, and years later it helped me in ways I could not have predicted. So for those of you who remember Animal House, this was the logo for Faber College. Knowledge is good. So one of the other take-home message here is knowledge is good. Learning things almost always pays dividends down the road, even if it can't always be predicted ahead of time. Okay, so I finished medical school, and I'm lucky enough to go to Johns Hopkins, again, thinking I'm going to be a clinician. I study internal medicine at Johns Hopkins. Here I am in the upper right. You can tell this is the first day of internship because all the white coats are on and they're all starched and neatly pressed. This is more like what the last day of internship looks like. The white coats are long gone. The tie, here I am, the ties are loosened. There's sort of a snarky inscription, again, another nod to Animal House. But importantly, during my intern year, my chairman was the great Victor McCusick, who was one of the fathers of modern human genetics, and he instilled in all the House officers an appreciation of the power of human genetics, as well as a sense of medical history. And that, as you'll see in a moment, had a big impact on me. And so another take-home message here is it's important to have heroes and role models, and one of mine was certainly Victor McCusick. So I was so convinced still that I was going to be a clinical doctor that I stayed an extra year at Johns Hopkins to be the so-called chief medical resident. Here I am with my fellow chiefs. I learned even more medicine during this year, and far more importantly, I also met my future wife, Kaelin, then Serbo, who was the mother of my children and my partner in life. But importantly, again, I learned a lot of medicine during this time. And amongst the things that chief residents like to know about are rare eponymous syndromes like von Hippel-Lindau disease, because when you're the chief resident, as the name suggests, you're supposed to be the alpha dog at all times. And so if one of the interns, or God forbid, one of the students challenges your authority, it's helpful to have knowledge of lots of obscure diseases that no one else has heard about. And then you simply can pepper them with questions about this disease they know nothing about until you've beaten them back into submission. And once again, you're the alpha dog. So what is von Hippel-Lindau disease? Well, it was first reported in the 19th century. It affects about 1 in 35,000 people around the world. It's inherited from generation to generation. It's a hereditary cancer syndrome where the patients with this disease develop cancers of various types. They develop blood vessel tumors called hemangioblastomas of the brain and retina. They develop kidney cancers. They also develop peripheral nervous system tumors called pheochromocytomas and a few others. Devastatingly, these tumors develop quite early in life, second, third, and fourth decades of life. And although it's improving, thankfully, the life expectancy, at least when we started, was only about 50 years. And I highlighted in that previous slide kidney cancer because hemangioblastomas and pheochromocytomas are very interesting, but admittedly pretty rare. Whereas kidney cancer is one of the 10 most common cancers in the developed world in both men and in women. And I mentioned that this disease is sort of inherited or passed down from generation to generation. Here are some family kindreds, or some kindreds of families with VHL disease that have been followed at the National Cancer Institute. So of course, each square is a man and each circle is a woman. And if the circular square is filled, that person has the stigmata of VHL disease. For example, they're developing those tumors. And so you can see how this disease is just passed from generation to generation, with about half of the individuals carrying the defective gene. And we've already talked about the classical tumors that are seen in this disorder. So I came to the Dana-Farber in 1987 to be a clinical oncology fellow. Amongst the Dana-Farber fellows, I was the one anointed most likely to go into clinical practice and the least likely to pursue laboratory research. But one of the paths to becoming board certified in oncology is to work in a laboratory for 2 years before getting your certification. And so it was clear that there was a molecular revolution in cancer that was just about to start or was starting. And so again, with some trepidation, I one more time decided I'd work in a laboratory. And I decided to work with a very bright pediatric oncologist, Shelley Bernstein, who had actually won a MacArthur Prize when he was younger, a so-called Genius Prize. And I just thought he was terrific. And so he was just starting his lab. So I actually was his first postdoctoral fellow. And then about 4 months after I joined his laboratory, Shelley said, you know, I don't know how to tell you this, but I can't do this anymore. I'm just going to shut down my laboratory and go into clinical practice. So this I took as a very bad sign. So now I was also his last postdoctoral fellow. And so now I'm thinking, well, not only am I not cut out to be a scientist, but potentially I'm capable of extinguishing other people's scientific career. So this is really bad. And what was equally bad was now I was an orphan. I just had to find a laboratory very quickly for the next 18 months. And fortunately, David Livingston took me in as a refugee. And now if in my first lab everything was wrong, and David's lab everything was right. And David's really the person who sculpted me into becoming a scientist, and he taught me innumerable things, but amongst the things he taught me as a scientist were to ask important questions and to be your own worst critic. And so despite my expectations, which were rather low, I actually did very well in David's laboratory. And so I think it's worth reflecting, why did I succeed? I've already hinted at one of them. I had great mentorship from David. I was given a great problem to work on. So the so-called retinoblastoma gene, a very important gene in cancer, had just been isolated, and David was working on this RB gene. And so I was one of the first people given the opportunity to work on the RB gene. Importantly, and this is important for the young people on the call, I was too naive to know what should not work. And hence I tried lots of things, some of which actually did work. And so it's important because as you get more experience, it's sometimes easy to talk yourself out of experiments. You know, there's always a reason why something won't work. But I think it's important to keep an open outlook. Good MDs are known how to multitask and to weigh conflicting data, and that's equally important in the laboratory. And most MDs are trained to be careful, and that likewise turns out to be very important in the laboratory. So now I'm going to digress for just 5 minutes. I know this is a very sophisticated audience, and this is probably unnecessary for most of you, but just to make sure we're all on the same page, I'm going to just demystify a little jargon that's important for telling you about what we did. So most of the time, when scientists use the word gene, they're referring to a segment of DNA that serves as the blueprint for making a protein. You probably know that DNA consists of 4 different building blocks, which scientists refer to with these different letters, A, G, C, T. You have about 20,000 genes. And for most genes, you have 2 copies, the one you got from mom and the one you got from dad. So the gene is the instructions for, as I said, usually for making a protein. So a protein is like a string of pearls that consists of now 20 different building blocks, which scientists refer to as amino acids, and they're represented with 20 different letters. Usually, but not all the time, the protein is a single strand of these beads that then folds into a 3-dimensional structure, which then determines its function. And so different proteins can perform different functions. So the logic of molecular biology really is that you have the DNA, which I've already told you sort of serves as the master blueprint, but you want to keep the blueprint safe. So if you want to make a protein, what you do is you make carbon copies of the blueprint in the form of RNA. So scientists refer to this as transcription, but you can think of this as like a Xerox machine that makes multiple copies of the instructions in the form of this transportable blueprint, which then gets sent to the factory floor to make a protein. Scientists refer to this as translation. So the order of these 4 letters, these bases, determines the order in which those amino acids get connected to one another. And this is, and one other piece of jargon. So sometimes scientists like to talk about genes being on or off, or highly expressed or lowly expressed. What they're really referring to is whether the Xerox machine is on or off, or is very active or less active. So again, we say that the gene is on if the Xerox machine is on. You're making lots of copies of the blueprint and you're making lots of protein, whereas in some cases, genes are off. And this is usually under the control of so-called transcription factors. Transcription factors are act as the conductors that turn on these genes. So for example, in your brain cells, the instructions for being a brain cell are being produced, but the instructions for making a liver cell are not on, they're not being produced. Now, why is this important? Well, even a single change in the DNA, such as shown here, can actually lead to the wrong amino acid being incorporated into the protein, and that in turn can lead to the protein not functioning properly. So actually here, I'm actually showing you a real mutation, and that's just a fancy word for an alteration. That is the difference between being normal or being, in this case, highly prone to some of those cancers I talked about, such as kidney cancer. And finally, when we, mutations in DNA are really critically important in cancer, because cancer arises only if a cell acquires the right combination of gene mutation. So I like to use the analogy of tumblers on a lock, where maybe this is mutation of gene A, and this is a mutation of gene B, and this is a mutation of gene C. And importantly, rarely, or sometimes, the first mutation is actually inherited from mom or dad. Okay, so now we can tell you what we did. So fortunately for me, right after I started my own laboratory, a group shown here, largely at the National Cancer Institute, isolated the gene that, when mutated or altered, causes that von Hippel-Lindau disease I told you about a moment ago. And just to make sure, and I should also just say in passing, and this will come as no surprise to the people at the GMGI, just to put this in historical context. So the VHL gene was cloned in 1993. This graph is simply to give you a flavor of the rate at which genes were then isolated and sequenced, and those DNA sequences were deposited in publicly available databases. So you can see that this was really the beginning of an explosion in genomics in terms of our ability to start to isolate and sequence and understand different genes. And so there's a saying in many cultures that can be reduced to, may you live in interesting times. So if you're interested in genetics and genomics, you can see why these have been such interesting times. So to make sure, again, we're on the same page in terms of the genetics. So in von Hippel-Lindau disease, the individuals have inherited a defective version of the VHL gene from mom or dad. So here I'm showing you schematically the maternal and the paternal copy of chromosome 3. So you may know that your DNA resides in chromosomes. You have 46 chromosomes. And here I'm showing you the maternal and the paternal copy of chromosome 3. The VHL gene lives on the so-called short arm of chromosome 3. So in this schematic, the individual has inherited an altered or defective version of the VHL gene from mom, but initially they're okay because they have a normal copy that they got from dad. But unfortunately, if you're born with a defective version of the VHL gene, you have about a 90% chance that some susceptible cell in your body, such as in the kidney or brain or eye, will spontaneously lose the remaining normal copy of the gene, and that's the cell that will then go on to form a tumor. And importantly, as you might predict from the knowledge that inheriting a defective version of the VHL gene can cause kidney cancer, if you now look at non-hereditary kidney cancer, and I'm sorry for this jargon, this is the most common form of kidney cancer is clear cell renal cell carcinoma, but we can just call it kidney cancer for tonight. If you now look at, off the street, non-hereditary kidney cancers, you again see that both copies of the VHL gene are missing or defective. But here, the individual started out with 2 normal copies, but sometime after conception, there was a mutation of the one copy and loss or mutation of the remaining copy. So this says that inactivation of the VHL gene is critical for developing some of these cancers, such as kidney cancer. And in the poor patients with VHL disease, they've already inherited the first defective copy. So how was my clinical training helpful? Again, knowledge, as I said, sometimes pays off in ways you couldn't have anticipated. Well, I knew from my clinical training, as well as that research I did in medical school, that the tumors linked to VHL disease are very rich in blood vessels. So if you, for example, take a patient with VHL disease and inject them with a fluorescein dye and look at their eye, you might find a retinal hemangioblastoma with the vessels filling with the dye here. And prior to CAT scanning, the diagnostic procedure in patients suspected of having a kidney cancer was to inject them with a dye and to look for the characteristic appearance of new blood vessels throughout the kidney. So that was sort of interesting. Another curiosity, again, chief residents love to memorize laundry lists like this. This is a so-called differential diagnosis, which for you and me is simply the causes and conditions, which in this case can cause excess red blood cell production, which is the opposite of anemia. Okay, anemia, too few red blood cells. These are the conditions that can cause you to make too many red blood cells. So some of these make sense, such as if you're living at high altitude or you have certain heart or lung problems. So making more red blood cells here will be adaptive, so you can increase your ability to capture oxygen. But some of these things are clearly maladaptive. Some tumors sometimes produce hormones that stimulate red blood cell production. And it always struck me as odd that the 3 tumors seen in VHL disease make this list, especially since hemangioblastomas and pheochromocytomas are otherwise relatively rare. So taken together, sort of the clinical aha was that VHL-associated tumors can stimulate blood vessel formation, and that's because they make a hormone called vascular endothelial growth factor, which is like a fertilizer for blood vessels. And they can also stimulate red blood cell production, and that's because they produce a hormone called erythropoietin. And what these two factors have in common is that they would normally be induced if a cell or tissue was not getting enough oxygen. This being a way to try to then improve oxygen delivery moving forward. And so it seemed like these tumors, the oxygen sensing pathway was defective. These tumors were acting 24/7 like they were starved of oxygen and were producing the molecular distress signals that you or I would produce if we weren't getting enough oxygen. And so that suggested there must be some link between the VHL protein and oxygen sensing. Now, the first experiment we did that showed that this was almost certainly true was in the laboratory, we created matched cells that had an intact version of the VHL protein or where the VHL protein was missing or defective. We grew the cells in chambers that had low oxygen or high oxygen. And then we measured the abundance of these so-called hypoxia or low oxygen inducible mRNAs, such as the mRNA for VEGF I told you about a moment ago. And remember, the mRNA is just the carbon copy of the DNA that's going to get sent to the factory floor to make the protein. So we knew that there were a number of these so-called hypoxia-inducible mRNAs. And as expected, if the cells had the VHL protein, these hypoxia-inducible mRNAs could only be readily detected under low oxygen conditions, hence their name. But if the VHL protein was missing or defective, now these cells merrily produced high levels of these hypoxia-inducible mRNAs, whether oxygen was actually available or not. So our guess was right, that the VHL protein was a critical part of the oxygen sensing pathway. Now, many of these so-called hypoxia-inducible mRNAs, or if you prefer, hypoxia-inducible genes such as VEGF and erythropoietin, are under the control of a transcription factor, and I introduced to you the idea of transcription factors. And in this case, the transcription factor is called hypoxia-inducible factor. Again, hypoxia being low oxygen. We knew from the work of many others that it contained a so-called alpha subunit and a beta subunit. But importantly, we knew that the alpha subunit was normally destroyed or degraded if oxygen was sufficient or plentiful. But if oxygen levels were low, now HIF alpha could accumulate, bind to its partner HIF beta, and turn on things like VEGF and EPO. And so our lab and several others went on to show, and this is frankly one of the most complicated slides I'm going to show you, so if we can make it through this, we're okay. So the VHL protein we showed binds to several other proteins, including these 4 proteins here. Their names don't matter. But what does matter is we and others showed that this multi-protein complex binds directly to the HIF alpha subunit when oxygen is present and attaches a little tail that scientists refer to as polyubiquitin. And that serves as a signal to send HIF alpha to a molecular garbage disposal called the proteasome. Whereas when oxygen levels are low or the VHL protein has been mutated, such as happens frequently in kidney cancer, now HIF alpha can accumulate, partner with HIF beta, and turn on genes like VEGF and erythropoietin. So this was very satisfying. Now we understood why cells lacking the VHL protein acted like they were deprived of oxygen 24/7. The other thing that was very gratifying here is I've drawn for you here the so-called alpha domain and the beta domain. In our biochemical studies, we could show that the alpha domain was critical for bringing in the rest of these protein partners, and the beta domain bound directly to HIF alpha. So it looked like these were the 2 business ends of the protein. And the reason this was so gratifying is we then solved the structure of the VHL protein with Charles Stebbins and Nicola Pavletić. So here I'm showing you the surface of the moon view, if you will, of the VHL protein. Here on the left is the alpha domain, and if we flip the protein on its side, here's the beta domain. And importantly, what I've done here is in yellow, orange, or red, I've colored amino acid building blocks that are the targets for mutations in VHL families around the world. So for example, one family might have a mutation involving this tyrosine at tyrosine 98. Maybe another family has a mutation involving this residue here. But you can clearly see that these mutations cluster in the so-called alpha domain and the beta domain, which we could now understand very well biochemically. Now, as you hope happens in science, having answered one question, we now raised another, and that was, how does the VHL protein know, if you will, whether oxygen is or is not available, and hence whether it should or should not destroy HIF alpha? And the answer, which we arrived at, and Sir Peter Ratcliffe arrived at working independently and in parallel, was in the presence of oxygen, an unusual chemical modification of one of those building blocks that we refer to as proline takes place. And in particular, one of two so-called proline residues gets hydroxylated. And once the proline is hydroxylated, the VHL protein can bind. And we understand this in exquisite structural detail, again, based on our collaboration with Nicola Pavletic. So here I'm showing you a somewhat different view of the VHL protein. We're going to focus specifically on the beta domain, which binds to the substrate. And the red you can think of as being sort of the VHL backbone skeleton. And the yellow are the side chains of those amino acids from the VHL protein that are looking for HIF. Whereas in blue are the region of HIF that actually gets recognized by the VHL protein. But the VHL protein will only recognize HIF if this proline has been hydroxylated, as shown here, because if the proline has been hydroxylated, these two fingers from the VHL protein can touch it and grab it. And so actually when Nicola solved this structure, he said he gasped because he realized if you were trying to design a protein that would distinguish between the non-hydroxylated proline and the hydroxylated proline, you couldn't do any better than this. Now, we and others then discovered the enzymes that actually attach this hydroxyl group. They're variably called the EGLN, or sometimes called PhD prolohydroxylases. What they do is quite elegant, and this is a tribute to nature, not us. They split the molecular oxygen, and then they use one of the oxygen atoms to hydroxylate the HIF alpha. And importantly, the enzymes have a relatively low affinity for oxygen, and so they're very sensitive to changes in oxygen availability over a physiologically relevant range. And just as an aside, perhaps of interest to some of the scientists on the call, these enzymes have several other unusual features in that they also require iron and a metabolite called 2-oxoglutarate. Now, importantly, since these enzymes do require 2-oxoglutarate, it immediately suggested that maybe you could modulate their function with drugs that would resemble 2-oxoglutarate, but otherwise inhibit these enzymes. I'll get to that in a minute, but I did want to point out that this system for oxygen sensing is conserved throughout metazoan evolution. So I'm sure we're going to have to now add the lobster here someplace. But what I'm showing you here schematically are the HIF genes. Here, the prolohydroxylases are referred to as the PhD genes, and here's the VHL gene. So in simpler organisms, there's a single HIF, a single prolohydroxylase, and a single VHL. As happened with many genes, there were some gene amplification events late during evolution that added some additional richness to the system. But this is the oxygen system that evolution has arrived at least if you're a metazoan. Now, I started to say a moment ago that in principle, you could modulate the function of these enzymes with drugs that would compete with 2-oxoglutarate. So here I'm showing you the structure of the major member of this family, which is variably called the EGLN1 or PhD prolohydroxylase. It has a nice druggable pocket. Even I can appreciate this is druggable because the scientists at Amgen who did this work are actually showing you one of their competitor compounds. And here I mean competitor from another pharmaceutical company in the pocket. And so we began a collaboration many years ago with a company called Fibrogen to develop these drugs initially for the treatment of anemia, because the idea here would be you would take this pill, your body would now think it's at high altitude, and you would start making your own red blood cells. This ultimately led to a drug called roxadustat. I'm showing you the two New England Journal papers recently that reported the positive phase 3 trials when these drugs were administered to patients who had chronic anemia related to chronic kidney disease. Frankly, an early indication that these drugs were going to be safe is that certain professional athletes figured out that they could get a hold of some of these chemicals from research chemical companies and were actually ingesting non-pharmaceutical grade versions of these compounds as a way of stimulating red blood cell production. But fortunately now the drugs have been through clinical trials. For a variety of reasons, the first approval took place in China, and here I am in Beijing watching roxadustat being made. But again, a number of these have advanced to late stage clinical testing, initially again for the treatment of anemia, but in time for the treatment hopefully of heart attack and strokes and other diseases where oxygen delivery is a problem. But I'm now going to change gears since I'm a cancer biologist and cancer doctor by training, and now talk about the role of HIF in at least those kidney cancers caused by VHL mutations. So I mentioned very briefly that HIF is also a member of a family. In humans, there's HIF-1 and HIF-2, and not shown HIF-3 alpha. But work we did showed that really the bad guy in kidney cancer is HIF-2 alpha, and if anything, HIF-1 alpha serves to constrain kidney cancer growth, and as a result, many kidney cancers simply silence HIF-1 and are exclusively producing HIF-2. So what can we do about this? Well, by the 90s, drug companies were fortunately already making drugs that block VEGF, and we've already talked about the critical role of VEGF in tumor angiogenesis, and we argued if these drugs were going to work against any solid tumor, they should work best in kidney cancer, where there's sort of a smoking gun because you have VHL loss leading to HIF, leading to VEGF. And fortunately, that's been, that was borne out in the clinic. We have now at least 7 VEGF inhibitors that are approved for the treatment of kidney cancer. They're shown here. So that's the good news. And some patients actually do well on these drugs for years. But the bad news is some patients either don't respond or they eventually will relapse on their VEGF inhibitors. So the question is, how could we do better? So you might say, just based on first principles, why target just one gene that's under HIF control? Why not target HIF itself? And based on what I told you, let's target HIF-2 alpha. Well, unfortunately, the conventional wisdom was that HIF-2 alpha was not druggable, that it lacked the right kind of pocket to be manipulated with a small molecule drug. But fortunately, Rick Buick and Kevin Gardner, who were then at UT Southwestern, ignored that, and they identified a potentially druggable pocket in HIF-2 alpha. I'm just showing you one portion of HIF-2 alpha here. And they identified chemical scaffolds that could bind to HIF-2 alpha, and in so doing, induce a change in the shape of HIF-2 alpha such that it could no longer bind to ARNT and hence no longer bind to DNA. And these compounds were then out-licensed to a biotech company called Peloton that was recently acquired by Merck. And the chemist at Peloton made these compounds more specific, more potent, and more bioavailable after oral administration. And they were kind enough to provide to us a so-called tool compound, PT2399, that was just 1 or 2 atoms removed from their lead compound at the time. And we showed that this compound did everything you wanted in laboratory models of kidney cancer. It could lower HIF-dependent mRNAs, such as the VEGF mRNA. It could stop kidney cancer cells from proliferating, for example, on a plastic dish, and could also prevent them from forming tumors in mouse models. And so eventually one of these drugs went into the clinic, and here I'm showing you data from the so-called phase 2 trial. These were patients with advanced kidney cancer who had failed standard of care agents, including those VEGF inhibitors we talked about a moment ago. And these are so-called swimmer's plots, where each horizontal bar is an individual patient and how long they stayed on therapy, at least at the time of this analysis. So for orientation, here's 1 year on therapy. So all the people with the black arrows were doing very well at the time of this analysis, still on study. And in fact, the patients with the gold stars actually had frank tumor shrinkage beyond a certain agreed upon threshold. So this was the glass half full. And based on this, this drug has now gone into phase 3 trials. But the glass half empty is that some of these patients didn't respond, and we're trying to understand this. Now, it's almost axiomatic in medical oncology that if you use drugs in end stages, in patients who have really been treated with many other things, have sort of been beaten up, they usually don't do as well as if you use the drug in a more frontline setting. And so here we got very lucky because the VHL patients are very well organized around the world. They have their own website. www.vhl.org. In fact, ironically, I discovered one day that the server for this website is actually in Brookline, Massachusetts, and I met a VHL family that started this whole process going. And now there are VHL patients communicating around the world about new therapies and what can be done. So we were able to convince Peloton to do a phase 2 trial in patients with von Hippel-Lindau disease who had never been treated medically before. So these patients had measurable kidney tumors that were just being watched in careful surveillance programs in an attempt to delay or prevent the need for repeated surgeries, because these patients develop so many tumors that you might be taking them back to the operating room very frequently. So these patients had never been treated medically, but they had measurable kidney tumors. And now when treated with a HIF-2 inhibitor, almost 87% of them had measurable tumor shrinkage. 40% had either a confirmed or awaiting confirmation, so-called partial response. That's a technicality we don't have to get into. Most of them were doing well and their disease had not progressed even after a year on therapy. And gratifyingly, although we were most focused on the kidney tumors, some of these patients had their brain and eye and pancreatic tumors, for example, shrink or go away. And so now here's what the swimmer's plot looks like for VHL disease. Again, here's 1 year on therapy. Now you can see most of the patients have a black arrow, are doing well on this therapy, which thankfully is pretty well tolerated. And many of them have also had objective tumor shrinkage. But statistics can be kind of dry. And so I want to put a human face on this. So for example, if you're as good at social media as my kids are, you could have found social media postings from some of these patients posting their results. And frankly, I can tell you that these patients have been living with the sort of Damocles over their neck for years. They've watched this disease, again, ravage their family generation after generation. And so it was very gratifying to see them posting things like, I never thought I'd see this day. And some of them are describing their tumors getting smaller, or in some cases disappearing altogether. Who doesn't like a video? So here's a short video.
Hey everybody, it's Justin. I just wanted to give you a quick update. I am in a gondola right now in Taiwan. Over there is Taipei 101. The gondola is actually right by the Taipei Zoo. But I just wanted to give you a quick update and say I'm doing well and enjoying my trip. If it wasn't for the PT2977 drug trial, I would have never been able to come out here and do what I'm doing right now. So I just want to thank Peloton, and I hope Merck will fast track this drug for a VHL treatment. So if you guys are listening, hopefully you guys will put it on the market to help VHL. But yeah, keep watching these videos. I'll be making more, and I'll get better at it, and I have to get the angles right because I kind of look fat, you know?
I love that, because at that age, you should be more worried about whether your vlog makes you look fat and not worried about what your next CAT scan looks like. So I'm just going to end with a couple of philosophical thoughts. So in hindsight, this is really the first paper describing von Hippel-Lindau disease. This was by the English geneticist Treacher Collins, and this was published in 1894, 10 years, frankly, before the paper by Eugen von Hippel. But ironically, one of several reasons why it became von Hippel-Lindau disease and why Collins' paper was overlooked was that this was the great descriptive era in medicine, the late 19th century and early 20th century. And so if you could only publish in one literature and you could only read one literature, you read the German literature. Now, Eugen von Hippel was a German ophthalmologist, so it wouldn't be surprising that he was publishing in German, but Arvid Lindau was a Swedish neuropathologist, and you can see that he published in German also just for the same reason. And I can tell you in the 70s, I studied German in high school. I was probably one of the last kids to be told that if you wanted to go into science or medicine, you had to study German. And in fact, when I was in medical school, one of my professors told me if I ever thought I discovered something new clinically, I should do a sabbatical and read the German medical literature, because the answer was probably already in the German medical literature. And I tell you that because, of course, there's no rule that says societies only continue to improve. They can regress. And of course, what happened with the horrific events surrounding World War II and the Third Reich, Germany's dominance in science and medicine evaporated. And frankly, they've never recovered. And in thinking back, of course, I started out talking about the 60s and how science and engineering were celebrated when I was a young man, but I do worry with some of the anti-science rhetoric that we increasingly hear, often coming from Washington. It sort of started with the war related to climate change, and then it became a general war on science and spilled over into the way we've approached COVID-19, and has led to this terribly disjointed response to COVID-19. So I do worry, as do many of my fellow scientists, that there are going to be lots of long-term unintended consequences of this really unnecessary and self-destructive war on science and truth. And at a time when we are maybe forgetting some of the lessons of the 60s, I can tell you other countries are not. In fact, other countries are trying to replicate what we did. Here's a picture from a lecture I gave in China in 2017. I purposely picked a lecture before I won the Nobel Prize because, of course, now when I've been to China, I need a bodyguard and it's selfie lines and autographs. But even in 2017, I was considered sort of a rock star. And here, I think every seat is taken except for these seats over here that I'm sure were reserved for government officials. In fact, to even get one of these chairs, the students had to undergo biometric scanning, including fingerprinting, just to prove who they were, to show that they were actually entitled to sit in on my lecture. So while countries like China are doubling down on science and engineering, I'm hearing language that really sometimes worries me in terms of the celebration of science and engineering in our own country. But to end on a happier note, again, for the young people, you may on occasion encounter someone who doesn't believe in you. You may on occasion get knocked down. You may on occasion be very disappointed. Again, I was disappointed with this. I was disappointed with this. But basically, you have two approaches. You can either wallow in self-pity. That's approach one. The alternative approach is to maybe use this as a little extra motivation. Maybe gives you a little extra fire in your belly to actually show these unbelievers that they were wrong. I would say the second is much more fun. So it was sort of nice when suddenly the Harvard Medical School Glee Club had to serenade me after I won the Nobel Prize, remembering my first interaction with Harvard Medical School wasn't quite so positive. This was, of course, a very gratifying moment in my life, but it was also bittersweet because I know at least a few of you know that my partner in life was my beloved wife, Dr. Kaelin, who was a very successful breast cancer surgeon at the Brigham, who developed breast cancer herself in 2003, but survived her breast cancer, but tragically developed an unrelated glioblastoma in 2010 that eventually took her life in 2015. So it was obviously painful not to share the Nobel Prize with her, but at least I was able to share it with my two wonderful children, Catherine and Tripp. And lastly, I started with this slide, and I want to also thank my parents. But I mentioned I had some of these toys, including this microscope. You may not know that when you win the Nobel Prize, one of the days you're in Stockholm, you go to the Nobel Museum and you make a donation. And I was the third of the laureates to make their donation. And I noticed my fellow laureates had donated things like raw data or notebooks or things that would have meant a lot to Mark Vidal and myself, but I wasn't sure were going to mean quite so much to maybe a 10 or 12-year-old boy or girl thinking about a career in science. So one of the things I donated was, in fact, that my microscope that my parents had given me way back when. So with that, I'll thank you very much for your attention. And if we have time, Mark, I'm happy to answer any questions.
These, the only thing that's missing in these Zoom events is the feeling of the people listening to you.
It is a little unnerving. Like, is anyone out there? Am I?
Yeah. And also, by the way, hey, Chris, some of the things you said were absolutely beautiful. Some of them funny. We can feel the emotion sometimes in your talk. I've known you a long time, and I really love that.
Thank you.
Anyway, Chris, we have many questions. So how much time do we have?
As much time as you'd like. I think everybody would really love to have you go through their questions. There's some really great ones in there. So
yeah.
maybe Bill, you could unshare the screen.
Yes, let's do that.
Okay.
Good. Good.
Yeah. So what I'm going to try to do is to take us first to in the science. Yeah, I mean, you have a couple of really good questions scientifically, and then it gets more philosophical, if that's okay with you to do it that way. So, for example, could some of the treatments that you described be used for COVID to help regulate oxygen in patients with hypoxia?
Yeah, so there's two elements to that question. Great question. So we can be sure that patients who have COVID pneumonia, who now have hypoxia in their lungs because there's a problem with oxygen delivery, are activating this very same system we talked about, that HIF is getting stabilized and HIF is turning on genes to try to promote adaptation to low oxygen. What I don't know is whether a little more HIF would be helpful. Alternatively, maybe HIF's in overdrive and maybe it's contributing to the problem. I simply don't know yet, so I'm not smart enough to know yet. And so my first rule is the body is usually smarter than we are. So before I would go in and start changing HIF pharmacologically, my working assumption would be that the adaptation mechanism is kind of working. But the reason it's a very astute question, in addition to that reason, is there's some soft preliminary epidemiological data that the risk of COVID decreases at higher altitude. So I'm working with some scientific colleagues to test in the laboratory whether having HIF activated actually helps protect you from COVID. And there's been, again, a suggestion that HIF might indirectly control the receptor for the virus. And so maybe this could start to make some sense.
Right. Yeah, that's great. Somewhat related, I like it too. Hypoxia promotes tumor growth, but causes tissue damage in heart attack and stroke. Can you comment on these conflicting roles?
Yeah, and I'll try to keep it, it's a very sophisticated question. I'll try to keep it.
It really is.
So in heart attack and stroke, the immediate problem is not enough oxygen, for sure. Once blood flow is reestablished, it is true that the sudden reappearance of oxygen can cause its own problems. And so that's why we and many others are now modeling in animals whether these pharmacological ways of activating HIF are a net good thing or a net bad thing. So far, it looks like it's a net good thing, but that will only be proven once we do human clinical trials. So in the heart attack and stroke situation, it's a little simpler. Now, in cancer, I'm sometimes asked, well, if you rev up HIF, isn't that going to promote tumor growth? Because you just told me HIF's part of the problem in kidney cancer. There are many levels to this question, but one of them is that tumblers in a lock analogy I used. There's really almost no evidence that HIF by itself can cause cancer. And frankly, VHL does some other cancer relevant things other than regulate HIF. So even in the worst case scenario, which might be the kidney, we know you need many other mutations to cooperate with VHL loss and HIF activation. But perhaps the better argument is we already know there are people around the world who, for example, live at high altitude, who have slightly elevated HIF, are making slightly more red blood cells, and they're not conspicuously cancer prone. So that suggests there's probably a therapeutic window where you can get the benefits of HIF without causing the dangers of HIF.
Great. And do you think some of the CRISPR technologies will be able to help VHL patients someday?
Yes, so just even stepping beyond CRISPR, I'm often asked, why not just replace the gene, correct the gene? I think that's all on people's blackboard is the place you would like to get. But as you know, we have some technological hurdles to overcome first. For example, it will almost certainly have to be 100% efficient, because if you only corrected the gene in 90% of the tumor cells, you could be sure that the 10% where you failed, those cells would simply take over and start growing again. So I think we just have to get to something that approximates 100% efficiency.
That's great. I love this one. Many marine animals, including marine mammals like whales, have a tremendous ability to adapt to low oxygen, for example, when they dive.
Yeah.
So do they use the same oxygen sensing pathway?
They are using this. It's a great question. They are using the same oxygen sensing pathway. But the reason I love the question is there are certain species that are really, really good, as the questioner suggests, at going to low oxygen environments. Another one is the so-called naked mole rat, which lives at very low oxygen. So I think understanding the genetic variants in these different species could be quite informative. But already we know, even in humans who live at high altitude, such as the Tibetans, they have very subtle variations in this pathway, genetic variations, that have allowed them to survive in these very low oxygen environments. So I'm sure there will be some very interesting genetic variants in some of these species.
Yeah, that's great. Somebody asked, I like this one too. Did you enjoy the challenge of working in the non-genomic era? And perhaps, and what, you know, was there any benefit to being?
Yeah, that's a great question.
Yes.
So I actually worry.
It's a black box. It's still a black box when you're doing this.
Here's what, I'll use an analogy. I once had dinner a couple of years ago with a fellow who was a retired nuclear engineer who used to design nuclear power plants. And he said to me, by the time we figure out that probably nuclear power done properly has to be part of the mix to get us off fossil fuel, the last person in the United States who ever would have designed a power plant would have long since retired. And I worry that the same might be true of biochemists, because we're in such a genomics or genetically dominated era that people want to identify the gene, sequence the gene, look for the variants. But in the end, to really solve the puzzle, you have to do some reductionist biology and some biochemistry. So I'm actually pretty glad that I trained as an old school reductionist biologist and learned a little bit of biochemistry. So, because I think you need both. I mean, genetics teaches you causation, but not mechanism. Biochemistry teaches you mechanism without causation, but if you can put the two together, it's pretty powerful.
So then we're sort of moving to less scientific questions, but more philosophical. One is from David Walt, one of our co-founders. So David says, thanks for the engaging talk. In the first part, it's a long question, but it's a great one. In the first part of your talk, when you described your path to science, although you started out being not a particularly good student, you ended up transforming yourself and you were able to be admitted to top colleges.
Yes.
So this is some of the young people on the call, as you know, our students at GBA, the academy, maybe are probably not going to be able to follow your footsteps, but what advice do you have for them with regard to achieving success and fulfillment in science?
I love this question. So first of all, I'm now ironically on the board of trustees at Duke University. And I pulled aside the dean of admissions, who had just shown us one application after another that were, stellar, perfect, straight A's, all 4 years of high school. And these were some of the kids who were not getting in. And so I pulled him aside. I said, would a late bloomer like me get into Duke today? And he said, sadly, you wouldn't have a chance, which I think is really tragic because, my view is, I've always said, I think if you're kind of bored in high school, yes, that might be a very bad sign, but it could be a very good sign. And maybe you just haven't found yourself yet, as I did. And also, when I see a late bloomer, I think, okay, maybe they've now achieved escape velocity, and maybe they even have another gear. Whereas the kids who have been operating at perfection for 12 years, often they don't always have another gear when they get to college and they're suddenly surrounded by, again, really terrific people. So I guess number one is just to acknowledge that reality. But I think more importantly, I'm a big believer in, don't peak early, keep your aspirations high. And I think the cream eventually rises. So you may not get into exactly the school you wanted or get exactly the mentor you wanted, but just do your best and hopefully the cream keeps rising. The last thing I would say is the most valuable thing I did, I think in terms of courses I chose, were mathematics, because things that teach you how to think never go out of style. Whereas a lot of other things you learn and memorize and regurgitate have a way of being forgotten or changing over time. So I think one thing I did accidentally get right was to study enough mathematics and computer science that it kind of prepared me when I did become a biologist to think in a certain way.
Two more questions, Chris. Is that okay with the timing? Hi, Dr. Kaelin. Could you speak to the nature of the partnership between the lab and the pharmaceutical company in medical research?
Yeah, so very briefly, I think academic labs should be in the knowledge generation business. And on occasion, if we generate enough knowledge, it creates opportunities to do things that are useful. And I think often the natural partners to do those things are our friends in biotech and pharma. So I think now there's, again, too much emphasis in the academic world on translate, translate, translate. But I think we should just be in the business of generating knowledge. And when you know enough, it becomes often obvious what you then need to do. But again, the people who have the skills to do it often are not in academic labs. They're often in pharma and biotech.
I love that. Also, before I ask you the last question, I just want to say that Alan and Bonnie are sending their warm wishes and hugs. Okay, you know who they are.
Of course.
And it's not their question, it's someone else. Last, what was the first thing you did after the call on that Monday morning?
The first thing I did was I took the fastest shower and shave I ever did. And I went downstairs and I started to fry an egg. And I've never eaten that egg because even before the egg was cooked, my cell phone started literally jumping off the table. My landline started ringing. There were Reuters and AP Press ringing my doorbell. And so the next thing I did was I frantically sent off a note to Lori Glemsher, the CEO of the Dana-Farber. I think it was like, help, 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 send support team. And so they sent up a SWAT team to help me with the communications.
Beautiful. So Bill, again, thank you very much. Chris is actually going to close the event. It was a pleasure as always. Thank you.
Thank you.
All right. Thank you both so much. Thank you, Mark, for making this happen and for your wonderful Q&A. And thank you, Bill, for so generously sharing your time, your insights, your stories with us. It was really amazing. We're so grateful. Thank you to the 9/11 Trust, James and Gail Bacon and New England Labs for sponsorship, and to everyone out there for joining in, for your questions and your encouragement. I'd like to also give a special thanks and a shout out to GMGI's Ashley Destino, who is the magic behind all the science hours, the newsletters, and so much more at GMGI. So we hope you're all going to come back in December and learn about Andrea's study of sea urchins, and please share the invitation with others whom you think would enjoy listening. We love coming into your living rooms and when we can't gather. And we hope that you have also received recently our 2020 annual appeal. And we hope that you've been inspired by these talks and by our work this year and all the years previously. And if so, that you will contribute and support GMGI's mission. Please stay tuned, stay in touch. We love hearing from you, and we really look forward to when we can gather again in person at our Institute on the Harbor. Thanks again, Mark and Bill, and good night, everyone.